So the last time we were together, we talked about the differences in the cell wall types of bacteria and why that was important. So somebody remind me very quickly, what's the differences between a gram-positive and a gram-negative cell wall? There are many ways to say that. Give me one way to differentiate them. What's the difference between gram-positive and gram-negative cell wall? Somebody tell me.
Color.
Okay, color is an easy way to get out of it,
I
Cheska.
know.
What about the cell wall type? What about the cell
Is
wall
it
type?
the coccus and bacillus
Gram
spirillum? No.
So those are morphologies. What about the composition? Jalen.
gram-negative bacteria are more resistant to antibodies,
That is true. That is true. But
or
it's
no?
not talking about
Okay.
composition. But what about composition?
Isn't
Jalen,
also the
there's a lot of
layers?
feedback. Huh? Okay, go ahead.
Isn't it also about the layers?
Yep, the
There's two
layers. Okay, tell
layers,
me about
and
the
one
layer.
of them.
Oh, in gram negatives, there's two layers, and so it has a smaller peptidoglycan and another layer. And what do we call the other layer?
The L. Oh wait, no.
The LPS.
Okay,
Very
okay,
nice,
that's what
Claudia.
it was.
And what is unique about the LPS?
Endotoxin.
It's endotoxic, right? By design. If it's gram negative, it's endotoxic. Very good. And therefore, the gram positive has a very large peptidoglycan, right? It also has these other things that anchor it to the plasma membrane. What are those other things, Cheska, that anchor the peptidoglycan in gram positive cells to the plasma membrane?
Oh, it was the taconic. I didn't say it that right.
Teichoic acids.
Teichoic
Yeah.
acids.
Very good. Good. The other thing we discussed was the differences in the glycocalyces, and we said that there were two types of glycocalyces, that there was a capsule and a slime layer, and they were both made out of carbohydrates. So structurally, they're a little bit similar, but they're a little bit different in the size of that external layer to the cell wall and functionality. And so what's the difference in functionality between a capsule and a slime layer? Kyung, what do you have to say about
Isn't
that?
it moving?
Okay, go ahead. Who was that? Cheska or Jalen? What's the difference
Yeah,
between
me.
a cap?
I thought it was
Okay,
movement,
one more time. I didn't hear you.
or no?
What was that? One more. You said what?
Movement, like
Not movement.
movement.
Not
I'm
movement.
sorry.
Not movement. Not movement.
Was it the layer?
Yeah, it's true, a layer, but what's the functionality between a capsule and a slime layer? What's their function? Remember, we're talking about these structures as they allow a competitive advantage for these organisms in an environment.
I'm trying to remember because I remember
The cell wall is protective, right?
Cell wall is protective, but we're looking at the glycocalyces. We're looking at the capsule and the slime layer. So which one of those, what's the functionality of those two?
associated with biofilms.
Okay, good. Which one? Claudia, which one? A slime layer. Okay, good.
Yes.
And that means that the slime layer is functionality is an attachment. Very good, Kayla. Thank you. So Kayla's written it really nicely, right? So Kayla says a capsule is about protection from phagocytosis. Very good. And then the slime layer is about attachment. Good. Perfect. Right. Those are the things I would like for you to have gotten and put into your knowledge bank already. Right. So today we're going to take it up with talking about bacterial appendages and why these different appendages allow these organisms, bacteria, to have an advantage in a competitive environment. So competitive environment could be anything. It could be a river, a stream, a brook, but also it could be the human body. And we're really interested in the competitive environment that we know as the human body, right? So we're going to talk about flagella and axial filaments. We're going to look at Fimbri and Pili, right? And then we'll talk about inclusions, and then we'll move into the eukaryotes. Okay, so let's go. So first of all, for bacteria, the flagella are the structures of locomotion. They're the structures of movement, right? They originate in the plasma membrane in prokaryotic cells, and they move like a propeller. So think about an airplane or a boat and their propeller and how they're moving, right? They move like this, right? And they have different forms, right? So structurally, they are attached and they originate from the plasma membrane. So you can see here, here's a gram-positive cell up here, right? You can see this is gram-positive here because it only has one layer, right? There's the cell wall, there's the plasma membrane, and here you can see how the flagella is hooked into the plasma membrane. Because the plasma membrane is the site of origination for the bacterial flagella, but it also powers the flagella. Okay? So if you look at gram-negative, again, right? So here's the cell wall, here's the LPS, and here's the smaller peptidoglycan, and here's the plasma membrane. And you can see that it originates, again, in the plasma membrane. And plasma membrane powers it, and then it has other structures that hook it into the cell wall. And now you have this flagella that's external, right? And it moves, you can see how it's moving. They're showing these little arrows moving it around so that it moves like this, right? Like a propeller, okay? So if we think about that, you can see that there are different types of arrangements, and here are some of those arrangements. Oh. So first of all, I want to make mention that this was meant to be a quiz, but most people get upset with me when I tell them they're not in order, right? So monotrichous is a flagella at one polar end, and it is represented by B, right? So you have monotrichous, you have one flagella, okay? Lophotrichous means you have a tuft of flagella at one polar end, and it's represented by C, right? Now it can have a tuft of flagella at one polar end, or it can have a tuft of flagella at both polar ends, right? They used to have different names, but now they have the same name. It's called Lophotrichous, okay? Amphitrichous means it's represented by D, that the organism has a single flagella at both polar ends, okay? And then peritrichous means that it's represented by A, means that it has a single flagella. I'm sorry, that means that it has flagella that surround the entire cell, right? So if I was to draw a peritrichous organism, right? Trichous means hair, peri means surrounding. So this is flagella that would be hair-like and be covering the entire cell of the bacteria, right? Just like that. There's one missing, and that one missing arrangement is called atrichous. And what does that mean? Somebody tell me what atrichous means.
No flagella?
No flagella. That's correct, Cindy. So if I was to draw an atrichous bacterium, it would just simply be the bacterium with no flagella. Okay, that means that they're not motile, right? We use the term motile to designate organisms that have flagella and can move by flagella. If they don't have flagella, then we say that they are non-motile. Right? So some people use the word mobile, M-O-B-I-L-E, but mobile means it can be picked up and moved from one place to another. Motile, when we talk about biological things, that means that they have the ability to move biologically, okay, and not physically. So if I pick up a pen and take the pen from one area of my desk and put it in another area of my desk, that pen is mobile, right? Mobile, right? But it's not motile. It cannot move by itself. Okay? Any questions? So this would be like a question on the exam next Thursday that'll just be fact-based. I would show you an image and I would have these 5 things: monotrichous, lophotrichous, amphitrichous, peritrichous, and atrichous. And then I would say what type of arrangement is being represented by the image above, and you would pick the correct one. Okay? That's just fact-based. Any questions? All right. So here is an electron micrograph. It's a SEM. It's a scanning electron micrograph, right? And why do we know that, Kayla? Why do we know this is a scanning electron micrograph? Why do we know that it's a scanning electron microscope, or we're using a scanning electron microscope? Anybody can answer?
Is it because of like the three-dimensional nature of
It's a
light?
three-dimensional structure, and we're looking at the surface or at the curvature, the topography of the cell, right? And so good. That's why that's because if it was a TM, we'd be looking inside the cell. Okay. So what is the arrangement of this particular organism? Can somebody tell me? What is the arrangement?
Is it Lophotrichus?
It's Lophotrichus, right? So you have a tuft of flagella, right, at one polar end, right? And so here you can see the flagella in a tuft out of the organism itself. Okay, that's Lophotrichus. Very good. So I just turned a fact-based question into an evaluation question, because you have to look at the structure, then you have to decide, oh, it's got a tuft of flagella at one polar end, therefore it's Lophotrichus. Okay, good. All right, now, anybody have any questions about flagella? There's a special type of flagella that we call an axofilament. Sometimes it's referred to as an endoflagella. As a matter of fact, it's probably used axofilament and endoflagella. They're probably used 50-50, right, depending on who you're talking to. But these are flagella that are associated with only spirochetes, right? And the reason that they're associated with spirochetes, do you remember when we were talking about the plasma membrane, and then we had just finished talking about the cell wall, and I said in bacteria there's this space in between the cell wall and this plasma membrane that's really important for cellular respiration, but also important for certain types of flagella. Does anybody remember what that space was called? Anybody remember what that space was called? It was called the periplasmic space, right? And so why that's important is because the endoflagella, or the axofilaments, are attached to a cell. Like, for instance, if I was to draw a cell, right, here is the cell wall. Here is the plasma membrane of a bacterium. An endoflagellum is attached at one polar end, and it transverses the cell in the periplasmic space and then comes out the other side, right? And there can be several of these that do that. Okay. And so if I was to draw that, attach that to this particular image down here, right, on the lower right-hand side of the screen, if I was to attach the flagella and then show it kind of transversing the entire cell and coming out the other end, and there's several of these that do that, right, you can see that in that periplasmic space, then you have these flagella. And tell me again, when flagella in bacteria actuate, how do they move? So what is the way that those flagella move? Right, somebody remind me. Remember, they move like
Plane?
a okay, not like a plane, but like the propeller on a plane, right? Good. So if the other images, right, this one is probably the best representation. You can see that endoflagellum wrapped around the plasma membrane, right? And here you can see this is a micrograph. This is a TEM. We're looking inside the cell, and now we can see those endoflagella wrapped around the periplasmic space, right? And so in order for something to be a spirochete, by definition, it has to be gram-negative, right? But also, in order to show the morphology of that tightly coiled, helically coiled organism, it has to have an axofilament. It has to have an endoflagella. In order to be a spirochete. Not a spirillum, but a spirochete. Okay? Any questions? Claudia, did you have a question?
Oh, no, no, no. I'm so sorry.
Okay. Anybody else? Questions? So
I do,
now you're saying, well,
Professor.
okay, go ahead.
You
Tamisha,
said
what's up?
in the beginning, you said this flagella was only associated with what? What was it?
With the spirochete.
Spirochete.
Let me write it down for you. Spirochete. Okay? So
Thank you.
remember, Tamisha, that if we are talking about spiral-shaped bacteria, they can have kind of like a shape like an S. They can have a shape like those birds you used to draw when you were a little kid. They can be bent at the middle. But a spirochete is very tightly and helically coiled, and it's pretty long, and it looks like this. Okay?
Okay, thank you.
Yes, ma'am. So not all spiral-shaped bacteria have an endoflagella, only spirochetes. So remember, in order for an organism to be a spiral-shaped bacterium, it has to be gram-negative, because in order to show its shape, the cell wall has to be flexible. And the gram-negative cell wall is flexible, but the gram-positive is not. It's rigid. Okay? Any questions? So you're asking, well, Provine, why is this important? Well, because all bacteria are attracted to nutrients, right? And so therefore, if they're attracted to nutrients, they're going to move toward nutrients, right? If they have flagella, they're going to move toward nutrients. Flagella are important because it allows for a bacterium to move to nutrients. And what that means is we have this word that we call chemotaxic or chemotaxis. And that means that the organism moves toward nutrients, right? So the bacterium move toward nutrients, right? Because they need nutrients in order to survive, right? And so they move toward nutrients, right, with their flagella, right? Now, some don't have it, and we'll talk about those in a minute, right? But for those organisms that have flagella, they exhibit a run and tumble type of format, right? Whether they have flagella in any type of arrangement, right? If we have monotrichus, amphitrichus, lophotrichus, peritrichus, or they have endoflagella, they're going to move toward, here it's calling it an attractant, right? It's just nutrients, right? But they run, and then they don't have any brakes, so they tumble, right? So they run and then they tumble, right? So here you can see that the flagella are here. They're actuated. They move like a propeller. They run, but then they don't have any brakes, so they tumble, right? So they're going to tumble until they run again, right? But they're going toward an attractant, and that attractant is nutrients. Yes, Jalen. Yes, Jalen.
Um, so these bacteria, are they the ones like on our skin as well? Like, are the ones on our
Mm-hmm.
skin motile,
They
or do
can
they
be.
have flagella?
They can
Okay.
have flagella, but they're not necessarily going to move all that much because there's not a lot of liquid, right, on your skin. These are going to
Yeah.
be much more found in your intestines, right, or in your mouth, those kinds of places, or uh, in your soup or whatever. Okay, good.
Okay.
Yeah. So here's the important thing, right? So we're going to talk about this in more. All bacteria are buoyant, right? They store gases in a vessel. So there's kind of just in whatever matrix they're in, they're just kind of floating like this, right? And then when they get the signal, there's nutrients, they move, all right, toward that. Because again, they're trying to survive, right? They don't think about it, right? They don't have that kind of consciousness, but they're trying to survive. Okay? Questions? Any questions? All right. So some other appendages are Fimbri and Pili, right? So you guys might see that in this organism right here, if you look real closely, you can see all these little hair-like projections that are coming off of a cell, right? They're coming off of the cell. I'm just kind of drawing them as they are drawn on here, but they're coming off the cell. And those things are found peritrichous to the cell, and they're called Fimbri, right? Fimbria for one, right? But they're all over the cell, right? Now, bacteria can have Fimbri, and then they can also have flagella, right? So the flagella can be lophotrichous back here, right? But the structures of those things are different, right? Where the flagella are the structures of locomotion, the Fimbri are structures that allow for attachment. And where we talked about, the slime layer and how it provided an advantage in a competitive environment because it allowed for the formation of biofilm and allowed them to stick to surfaces in each other. These are even more important because the organisms, the bacteria that are fimbriated, that have fimbria, can attach to our cells and our tissues, right? So here is a micrograph, right? It's a scanning electron micrograph that's been a little bit enhanced, but you can see the Fimbri surrounding them completely, right? What is the arrangement of these cells here? Somebody tell me. What is the arrangement of those cells?
Is it also a lophotrichus?
Oh, so you can't, so these are going to be peritrichous because they're Fimbria. They're not flagella, right? But what's the morphological arrangement of those cells, Cindy? Not about the Fimbri. The Fimbri are peritrichous. But what about these organisms and a
Is
pair?
this regarding like gram negative and gram positive?
No, not necessarily. It's really regarding just the arrangement, right?
Okay.
It's regarding how these things are arranged, right? There are two of them together, right? What
Oh,
do we call those?
Coxy?
So that's the shape, right? But then what's the prefix that describes a pair?
Is it budding?
Mitosis.
Not
amorphous?
budding, not amorphous, diplo. That's right, Giovanni, that's correct. So these are diplococcyi, okay? So this is an actual micrograph of Neisseria gonorrhea, right? This is what causes gonorrhea, sexually transmitted infection. And so you contract this by having sex with somebody who has gonorrhea, right? So you can see these fimbria, this organism, right? And the most dangerous, the most virulent forms of Neisseria gonorrhea are fimbriated, right? Because if they make contact with the cells of the tissues of the penis, inside of the penis, the inside of the vagina, or really, with females or with males, the tissues of the urethra, then you can develop infections there, right? So after sex, and you might not know the person very well, but after sex, you should, after a few moments of cuddling, you should both get up and go and urinate, right? So that if there is gonorrhea present in one of the bodies, then you flush it out before they establish themselves, right? Because once they establish themselves and they are fimbriated, then they're hard to get rid of, right? And so you might be saying, well, Pro V, the vagina and the urethra in females is different piping. That's true. But what we know is that females who develop gonorrhea intravaginally, a lot of times also develop gonorrhea in the urethra, right? So we don't want to get both of these particular sites infected with gonorrhea. All right. So you can see the importance of the fimbria, right? Because even if they've established themselves and even after you urinate, they're not going to be dislodged once they're embedded in the tissues, and then you're going to have to be treated with antibiotics to get rid of gonorrhea. Okay? Questions? Anybody have any questions? All right. Let's go on. So here you can see that this particular organism has the fimbria, right? These are really small projections that are used in attachment, but they also have flagella. Okay? When we talk about pili, it's a little bit different, right? So here you can see two different cells being attached to each other by a pili, right? So if I was to draw a cell, these are gram-negative only. If they have pili, then there are these structures that are much more substantial than a fimbria, right? And they can attach to other cells like this, other bacterial cells, right? And they have to be gram-negative, right? Because this is only a gram-negative mechanism, right? So here you can see that this one cell right here, this one right here, is attached here to this cell. It's attached here to this cell, and it's attached to here in this cell, right? So you're saying, well, why is that important? Well, because let's say, oh, by the way, these in this image, there are two different species. How do we know that? How do we know that there are two different species?
The Fimbria is on the outside, and then the other one doesn't have it.
They both, oh, okay, I see what you're saying. So you have these fimbria here, right? Good, and this one doesn't have it. So yeah, morphologically, they don't look the same, so they are different, right? But let's say that both of these cells have chromosomal DNA, right? And the chromosomal DNA is in the cell. And they're different, of course, but one of them has what we call a plasmid, right? And plasmids are small pieces of DNA that are extra chromosomal, not part of the chromosome, and they're self-replicating, and they code for the resistance to antibiotics or for the production of a specific metabolic end product, like a pigment or like a toxin, okay? So now this cell, whatever it is, the J-lin cell, can donate that plasmid, make a copy of it, and donate it to this other cell, the Cheska cell. And now both of these cells have the plasmid that basically says they are resistant to penicillin, right? And so this is one of the mechanisms that gram-negatives have to become resistant to antibiotics. There are a lot of others, right? We'll be talking about some of those others, but this is one mechanism that they have to be able to resist antibiotics. Isn't that interesting? Any questions?
How do you know that by looking at that?
What do you mean?
Well, because wasn't that what you were asking us? Like, how do we know what else it could be? And is that how you?
So wait, okay, let me go back. So now tell me what you mean by that.
Well, because
Ask,
you asked us like what was the difference besides the, the
oh, okay.
obvious,
So
right?
I think Jay-Lin told me that
Okay.
they were different because this one had visible fimbri, right?
Got it. Yes.
But this one doesn't, right there. So they're different.
Okay. Okay.
And the fact that they have a pili, right, means that they are undergoing what we call conjugation, right? That means that they're transferring some DNA, plasmid DNA, from one of the cells to another. Okay?
Got it. Cool. Got it.
Okay. Anybody
Could
else?
could similar cells or I guess not similar cells or like same species of bacteria, can they do this
Yes.
amongst each other?
As long
Okay.
as long as they're gram-negative. Only gram-negative cells do this.
Okay.
Okay? Gram-positives do not do this. There's been one paper written that has proposed that this is a gram-positive mechanism also, but there's not enough evidence that even gets close to saying, yeah. So it's a gram-negative mechanism only. Okay? Any questions? So, again, it's about one organism being the donor, and the other organism being the recipient, right? And the plasmid gets shared from the donor to the recipient. Okay? Again, this is a process that allows these organisms to survive in a competitive environment because it allows the sharing of plasmids, which means that it makes those cells harder to kill, right? Because now they have the resistance to an antibiotic. And it's not just penicillin. There's a lot of resistance out there to lots of different antibiotics. And almost all of them are plasmid mediated. That means that they do that because of a plasmid. Okay? Questions. And you spell plasmid like this. Okay? Any questions? All right. So we've talked now about all the important structures on the cell, right? We're going to look now at ways that bacteria store things, right? And we call these inclusion bodies. They're vacuoles, right? So if you think about a bacterial cell, right? They might have DNA, right? And that's considered an inclusion. It's we call that a nucleate, but it might also have inclusions in it that might store lipids. It might have, and these are going to be vessels, right? They're going to be vacuoles, just storage vacuoles. They might have another one that stores phosphates, right? They might have another one that stores glycogen, right? So each of these vacuoles or these vessels that are going to store different things, okay? And that's what these inclusions are, right? So they don't have organelles, but they do have storage vacuoles. They do have ways to store things, okay? The first one is called polybeta hydroxybutyric acid. And this is the way bacteria store lipids. Now they store lipids mostly for the formation of new plasma membrane, the only membrane they have, during binary fission from one cell becoming two cells. They're going to need to build more membranes so that each of the cells have a complete cell, okay? If they get into rough times, they could also use some of these lipids for nutrition, right, to survive. But mostly it's because they are building membrane, okay? Bacteria store glycogen, right? So glycogen, it's a storage, right? It's a storage vacuole, so there should be a comma here. It's a storage, and it stores glucose, right? So it's like starch, right? But it stores glucose. And the bacteria store it to be able to use that. If they run low on glucose, they can use the glycogen, break it down enzymatically to a whole bunch of glucose molecules, and then they can use it for the production of ATP. We also, as animals, store glycogen. So anatomy and physiology, you should have learned this. Where do we store glycogen? Where do we store glycogen as humans? In the liver, right? That's right, Carolina. We store most of our glycogen in our liver, and we do store a little bit associated with the muscle tissues, right? But most of the glycogen we store is in our liver. And so if you haven't eaten breakfast yet, you are depleting your glycogen levels pretty quickly. And around another 2 hours or so, if you still haven't eaten anything, there's going to be a physiological change that happens in your body. Some of us get shaky, our hands start to shake a little bit, right? Some of us get loopy, right? 2 + 2 is 7, you get loopy. Some of us get kind of, we become kind of grumpy people, right? We say people are hangry, right? They're hungry and they're angry because they haven't eaten yet. But those are all physiological changes, right, that happen in the body, right? They're all done by chemical signaling, right? Because we're much more complex than a bacterial cell, right? And so if that happens, then we go into what we call a ketosis, right? And so our cells go into a ketosis. Bacterial cells can go into a ketosis also, but not as readily, right? We go into ketosis because we're a much bigger organism and we use up all our glycogen. And then a couple things happen. Our breath can become really, really bad smelling, right? You can always tell, at least I can, when a person is under one of those fad diets, like the keto diet or the Neanderthal diet or the caveman diet or the protein diet or whatever, right? Because they're not eating any carbohydrates. They're just eating protein and fat. And the body itself needs carbohydrates all the time. And so if it doesn't have it, they're going to break down the next important macromolecule that they can break down, right? And so remind me from anatomy and physiology, 2401, what are the four macromolecules that you all should know already? What are the four macromolecules?
Lipids,
Lipids,
amino
correct?
acids.
Not amino acids,
Um,
but amino acids make
um,
protein. So lipids and protein, that's two of them. What are the other two? Lipids and
glucose.
protein.
Or no,
Carbohydrates and
carbohydrates.
fats.
Carbohydrates, right? Glucose is a form of carbohydrate, right? So a type of carbohydrate. And then the last one is what? The hardest one. Nobody ever thinks about it because none of us eat it or consume it as a nutrient, but it's important, which is the nucleic acids. Very good, very good. So the four macromolecules are lipids, proteins, carbohydrates, and nucleic acids. Very good. So if our body doesn't have any glucose, right, after a certain amount of time, then the body will attack the next macromolecule that's available to it. And what is that? What is that?
Lipids?
Not lipids. The body does not attack lipids. Well, but Kayla, true, carbs are its favorite source of nutrients, but if there are no carbs, then what does it attack? So
Protein.
protein. So it breaks down protein. Therefore, you go into a ketosis because the breakdown of the amino acids leads to ketones, right? And a lot of ketones in your body is not good, right? So if you are releasing ketones from the body in the urine, right, or they find a lot of ketones in your blood, that means you're starving, and that's not a good physiological means. The body will only break down lipids in the presence of carbohydrates. So if you want to lose weight, you have to cut down on the amount of carbohydrates that you're consuming, but you still have to consume carbohydrates, right? So cut down on the donuts, on the sodas, and those kind of things, and only consume things like fruits and vegetables, or some of the bitter carbs for you, like pastas and things like that, right? But stay away from donuts and cakes and cookies. It's hard to do so, but yeah. So we really are pretty similar to bacteria, but other organisms like the fungi and the plants also have storage, right? They store different types of things, right? But it's interesting to see the similarities of how we store things and how the bacteria store things, right? Number three, bacteria store phosphates in what we call or polyphosphate granules, sometimes referred to as metachromatic granules, right? Because of the way they stain under the microscope. But this is the storage of phosphates, and they store phosphates to make more membrane, right? Plasma membrane, but also for the formation of ATP from ADP, mostly in the electron transport system. Okay. Um, Sulfur, they store sulfur, and they store sulfur because they need it to make some different amino acids if they have to make them, right? Which make them proteins. And so with the proteins, the sulfur is really important also because if you have this chain of amino acids, right? If you have these chains of amino acids, and it's a long chain of amino acids, the disulfide bridges will form in between the the chains of amino acids, right? And these disulfide bridges, right? These are sulfur containing, right? Form these bridges, right? So you have sulfur attached to sulfur. And what that does is it helps the protein start to fold on itself, right? So the protein folds on itself, and it starts to take shape. And you guys learned in Anatomy and Physiology 1 that the shape of the protein establishes its function, right? So if this is the shape of the protein, then it's going to function in a certain way, right? This is the binding site of that protein, or the active site of that protein. And remember, so you can see how all these things are important. Now, bacteria have the same mechanisms we do, right? They just are more simple in the way they're designed, but we have very complicated systems. But the process of folding these proteins works the same way. And we'll talk about this when we look at transcription and translation later in the semester. Okay? Any questions? All right. Gas vacuoles. So they store metabolic gases, right? So that they can be buoyant, right? And so they are allowed to float in the fluid matrix that they're in. Because if they didn't, they would just sink to the bottom, and then they wouldn't get any nutrients and they would die. So here, if they're floating and they have flagella, then they can move toward the attractant. But if they don't have, if they're atrichous and they're floating in the fluid matrix and something bumps into them, or the matrix pushes them into something, and that something might be an attractant, it might be a nutrient, then they can break down that nutrient, right? So those bacteria need to be buoyant in order to be able to survive. Ribosomes, again, are free floating in the cytoplasm, right? So different than in eukaryotes. But if you have a bacterial cell, and here is the DNA, right? Not in the nucleus, but the ribosomes are going to be free floating in the cytoplasm, right? And they can form chains and make a whole bunch of proteins all at once, right? And what we call a polyribosome. And that's pretty cool to think about, how sometimes more simple is more efficient and more effective. Okay. The nucleoid material is the DNA, the chromosome of the bacterium. They're one chromosome. They only have one, right? Oid means resemble. So if you are a humanoid, you resemble a human, right? So nucleoid means it resembles a nucleus, but it's not a nucleus. Some people would confuse it if they don't know any different, because a lot of times the DNA in bacteria is kind of in one area, and if you're not knowing exactly what you're looking at, you might think it's a nucleus, but it's not, right? Bacteria do not have a nucleus, right? And then we talked about plasmids, right? So here's a definition of plasma. It's a small fragment of self-replicating extrachromosomal, not part of the DNA, that codes for resistance to antibiotics or for the production of certain different metabolic end products like pigments or types of toxins, right? Good. Sometimes this plasmid can jump into the chromosome. It can enzymatically be put into the chromosome, and that leads to a mutation, right? Mutation means there is an inheritable change that happens in the DNA molecule, and that is almost always good for the bacteria, because that's the way that they become a different organism, right? They don't undergo any kind of true sex, right? So if we think about that, every time we have progeny, every time we have kids, each of our kids is different genetically, because there's half of the chromosomes from the male and half of the chromosomes from the female. And so what you have then is you have a genetically unique organism, right? Bacteria are a bunch of clones of each other, right? So the only way there can be any kind of change in their cells is for mutations to occur. And every 10 to the 7th division, every millionth division of a bacterial cell, there's mutations. So we have millions of mutations going on every day, right? And we just don't see them. Okay? Questions? That's why I always say bacteria are the great survivors, right? We as humans may drive ourselves off the planet of the Earth, off the planet of Earth, but the bacteria will still be here. Okay? So here is kind of distinction between the chromosomal DNA and then you see the plasmid DNA. Remember, the plasmid DNA is small, it's self-replicating, and it's extra chromosomal, right? It's away from the chromosome. Okay. This next image shows that the plasmids, every time the cell replicates, that each of the new cells get a copy of that plasmid, right? And then here it shows that sometimes the plasmid can be integrated, become part of the chromosome, and that leads to a mutation, which then simply means that it adds to the diversity of the bacterial population, right? It changes the bacterial population. Any questions? Okay. So here's a
I
beautiful,
do.
yes, ma'am. What's up?
So if bacteria can change, like, how do we know what specific like bacterias are which? Like, if they're constantly changing and like doing
You won't
everything,
be
how
able
do
to.
we know?
You won't be able to know unless you're actually studying them at a molecular level. Or if all of a sudden, um, sometimes this happens. So sometimes, you're taking like my generation, Jalen, grew up where my very wealthy friends. It was not me. I did not grow up wealthy, but my wealthy friends. I went to a high school that was pretty wealthy, but my neighborhood was not. But my wealthy friends, a lot of my friends that were female were taking antibiotics for a long time, and so they always had beautiful skin, right? Because the antibiotics they took wouldn't allow the bacteria to cause pimples and things like that, right? But every once in a while, one of my friends would develop an infection, and the infection would be a bacterium that was hard to get rid of because it had developed resistance to the antibiotics that she had been taking for a long time, right? So you're not going to know unless you're really paying attention or unless you're doing molecular work with looking at the DNA sequences of bacteria. You're just not going to know. It's just going to be part of life. You're just not going to really think about it. Okay. What's the gram specificity of the cells you're looking at in this particular field of view on the slide? What's the gram specificity?
Pili?
What, what was that? What was that? I heard kiddos in the background. That's all I heard. So, what's the gram specificity?
I think she said the pili.
One more time.
I think she said the pili.
Oh, these are Pili for sure. But what's the gram specificity? The answer is so the choices are gram positive, gram negative. What's the gram specificity?
It's gonna be gram negative.
It's going to be gram negative because only gram negative have Pili. Okay, good. Questions? All right, that's a beautiful image right there, right? That's a nice microscope work and taking of pictures. Okay, the last inclusion body we're going to talk about is an endospore, right? So endospores are only produced by gram-positive bacilli. Right? So if you see an endospore in a bacterium, by definition, it is a gram-positive bacilli. And there are lots of different genera that produce it, but the two that are clinically relevant are Clostridium and Bacillus. Clostridium has a lot of species in it that produce problems in us, diseases or infections, right? And these are some of them, right? They produce things like tetanus and botulism and antibiotic-associated diarrhea, C. diff, right? They produce things like gas gangrene. They produce things like food poisoning, right? Bacillus is less dangerous, but there are some really important pathogens in that group. So Bacillus anthracis causes anthrax. Bacillus cereus thuringiensis produces food poisoning, especially in high carbohydrate dishes like rice or fideo or vermicelli or whatever, right? So those organisms are really important, right? The endospores are a survival mechanism for the bacteria that are gram positive. Not all gram positive bacteria have them. not all gram-positive bacilli have them. The two genera that are most important to us from a clinical perspective, from a medical perspective, are Clostridium and Bacillus, right? It's a survival mechanism, mostly because the outer covering of an endospore is composed of dipicoic acid and calcium. And that makes these endospores resistant to heat and desiccation and freezing and all kinds of chemicals, right? So if you are boiling something, you can get rid of all the vegetative structures. You can get rid of all the bacteria, but you're not going to be able to get rid of the endospores. The only way the endospores can be destroyed is by very high temperatures that are pressurized, right? So an autoclave. That's the only way. You're not, or radiation, but you're not going to be able to destroy them with just boiling. Okay? And so what happens is one bacterial cell that is a gram-positive rod that can produce endospores, about every 30 minutes, one of these cells will become two, right? So every 30 minutes, one of these clinically important bacteria become two. If the bacterium starts to get into really rough times, right? No nutrients stress. To bacteria, stress is no nutrients. They will start to form an endospore. And an endospore is a way that they produce this vacuole, this vesicle that stores the DNA, right? And so these continue on until essentially the bacterial cell is completely engulfed by it, by this endospore. And eventually what happens is that the parent cell dies, but what's left over is the endospore. And the endospore can remain in the environment for years, right? Hundreds of years. I have an article that I read about a mummy that was found in Peru, and they found endospores on the mummy. And now they're studying those endospores to see what organism they are, right? Hopefully it's not what killed the mummy, right? Because then we might unleash some really dangerous organism, right? But those endospores can remain dormant in the environment for a long time. And then what happens is maybe the environment becomes hospitable again, in that there's nutrients back in the environment. And what happens is that the endospore germinates. That means it opens up like a seed would. And what it does is that DNA that was in the endospore forms an exact copy of the cell that produced the endospore to begin with, right? And so it's a really, no, herpes is a virus, right? But herpes does go dormant, right? And so you can contract herpes, and then herpes can develop lesions on the urogenital area or on the buccal cavity, right? We call them cold sores. And then it go dormant, but then you get stressed out. And when you get stressed out, the virus starts to replicate again. And then you have problems with outbreak of herpes again. No, this is different. So this is a bacterial mechanism only, and a gram-positive bacillus mechanism only, for Clostridium and Bacillus only. Those are important clinical organisms, right? They cause all kinds of problems in us. Okay? Any questions? So how many people have heard that if you get stuck? Cindy, what's up?
Sorry, can you tell me what desiccation is again?
Desiccation means drying out.
Okay, thank you.
Okay, yeah. So anybody ever heard that if you get stuck with a rusty nail, you're going to get tetanus? How many people have heard that?
I've heard that before.
Yeah, but that's not true, right? The rusty nail, that implies, that statement, if you get stuck with a rusty nail, you're going to get tetanus. That implies that tetanus is caused by a rusty nail. That's not true. Tetanus is caused by a toxin that's produced by Clostridium tetani, right? So here's the organism's name. Clostridium tetani, right? It's endosporic, right? So the endospores are in the soil, and the rusty nail has got soil in it, so it's got endospores. So you get stuck by a rusty nail, and then the rusty nail allows for the injection, the inoculation of the endospores into your tissues. It begins to grow, and then it can cause tetanus. Tetanus, when we have tetanus, it's not the bacteria that's causing this problem. It's a toxin, right? The toxin is a neurotoxin. It produces kinesiospasms, and that means that your neurons are constantly firing. And so you have these huge contractions of the muscles, which have been known in some cases to break the larger bones of the body, right? Um, the thigh bone or the rib or something like that, right? Because of the contractions, right? But that doesn't mean it's just a rusty nail. It's any time that you get a laceration, a cut, or a puncture wound that's out there in nature, right? And so that can be from a piece of glass. It can be from a piece of wood that you get this kind of splinter in you and it gets punctured into you. It can be that you're skateboarding or bicycling and you have an accident and you have road rash, right? All these different ways is the way that the tetanus endospore can enter the body and then cause problems. So a lot of times when you get a cut or a laceration or a deep puncture wound and you go to the physician to get stitched up or cleaned up or whatever, they'll say, we'd like to give you a tetanus toxoid vaccine, right? You should always say yes because it's not going to hurt you. But toxoid means resembles a toxin. So what we've done is we've denatured the toxin so it doesn't do the things the toxin does, but it still looks like the toxin, a protein, and then the body can produce antibodies against it. And if you start to develop the infection which causes the toxin, the antibodies against the toxin will not allow it to cause tetanus in you. Okay? Good. It is one of the few, there are a couple others, but it's one of the few vaccines that we have against the toxin and not against the bacteria. Okay? Questions? Okay. So again, here's that cycle that I drew for you, although this is probably a lot better, right? So it shows the cycle of the parent cell producing the endospore taking over and eventually being released into the environment, right? And then that can germinate and form an exact copy of that bacterial cell, that vegetative cell that formed it to begin with. Okay? Here's an electron micrograph showing the endospore. It's pretty nice. And if we get to go back to the classroom, we're going to do endospore stains, and you're going to be able to see the endospores inside of the bacteria, right? So the green structures inside the bacteria, the vegetative cells of the bacterium are staining red, but the green structures are the endospore. Okay? Any questions? All right. So I am done with prokaryotes. We're going to talk about eukaryotes now, and I'm going to do this pretty fast because you guys should have already heard a lot of this in Anatomy and Physiology 1, right? So I can do quick comparisons now, right? And so if you took 1308, even better, because I know people like Cynthia and other folks who took me for 1308, or Amanda, right? You had this already for me, but you should have also had it in 2401. You should have had it in Anatomy and Physiology. So I can go through it real quickly, right? So let's talk about cell walls first, right? So think about the cell walls of bacteria, right? They're mostly composed of peptidoglycan if they're gram positive, and then if they're gram negative, they have peptidoglycan carbohydrates and an outer membrane called LPS. The organisms that are eukaryotic that have cell walls are going to be plants, they're going to be fungi, molds, or they're going to be algae, right? So plants have their main sugar, of course, is cellulose. We as humans do not have the ability to digest cellulose. So if you eat plant material, then you can see it in the feces because it comes out like the plant material, right? It's a little bit beat up because it's been through heat and all kinds of enzymes, but the enzymes weren't able to break it down, so out it comes, right? So if you have eaten corn, you think you chewed up all the corn, and there they are, whole kernels, right? Yeah. Anything that has a lot of cellulose in it, we cannot break apart, right? So plants, right? This is a fungus. Does anybody know what the name of this fungus is? You should, because we studied it in lab. What is it? Looks like a brush or a broom or a hand. What is it? Anybody know?
Penicillium?
Penicillium. So the fungal cells, their cell walls are made up of cellulose also, but they can have chitin in them, right? So chitin is another type of long chain carbohydrate, right? And you mostly are going to find that in the exoskeletons of arthropods like insects or whatever, right? But also you can see that they make up some of the composition in the fungi. And then the algae are composed of cellulose, but they also have silica and calcium carbonate in their cell walls because of the aquatic matrices that they live in, right? But these are the major components of the cell walls. They're not really microbiological related, right? So if I ask any question, it's probably going to be around the mold, right? I might say something uh, like a multiple choice question would be, the major constituents of the mold cell walls are cellulose and a blank, and then it'd be A, glycogen, B, uh, starch, C, glucose, D would be things like chitin, and that would be the answer. Okay. Questions. Rebecca, did you have a question? Okay. Plasma membrane. Right. The plasma membrane structurally is very similar to the prokaryotic membrane, right? They're composed of phospholipid bilayer. They have integral proteins associated with them, right? They have glycolipids, got glycoproteins, right? But their function is mostly in the regulation of what comes in as a nutrient and what leaves as um, a waste. It's the same rules apply, right? If they're going to come directly into the membrane, they have to be small, they have to be uncharged, or they have to be lipophilic. Anything else, including water, has to come through a pore, right? The one difference that we see between our cells, animal cells, and the bacterial cell is the bacterial cell have cell walls, and so do plants and algae and fungi, mold. And so the cell walls really keep the plasma membrane in place and keep it from breaking apart. Animal cells run into the problem if it gets too warm, the plasma membrane starts to break apart and our cells will break apart, or if it gets too cold, they can become more rigid and then they don't function properly, right? So the way that evolutionary that we have dealt with this, that our cells have dealt with this, is that there is this particular molecule that is in the plasma membrane of animal cells. It's this little yellow structure right here that is embedded in the lipid tails of the phospholipid bilayer. And what is that, Miss Amanda?
Cholesterol?
Cholesterol. Beautiful job, Cindy. Beautiful job. So cholesterol, and cholesterol functions in animal plasma membranes, cell membranes, to keep it stable, keep it from breaking apart. Okay. Questions. Structurally same, functionally only regulates what comes into the cell, leaves the cell, right? It is involved in signaling a lot more because our cells are much more complicated, but that's an anatomy and physiology 2 mechanism that you guys are going to be talking about, right? It's a little bit outside the scope of the microbiological perspective. So you'll be dealing with cell signaling in anatomy and physiology 2. Okay. Questions? All right. Glycocalyces, right? So capsules mostly, and only for a few types of cells like the yeast. The yeast do have capsules, some of them, and they also are made out of sugar, carbohydrates, and they are responsible for protecting the yeast from being destroyed by phagocytosis inside of us, right? They could actually be also a nutrient reservoir, so like the bacteria. But the eukaryotic cells are so much more complicated, and they're so much more complex, and they have all these other structures inside of them that can help them survive, right? They don't need to have a capsule, right? They have all these different enzymes that can be released. You know, there's so many things going on that they don't really need capsules. Yeast are more closely related to the bacteria, right? And so they do have a capsule, okay? And it serves to protect them. Any questions? All right. Now we get to appendages, right? And so appendages, we're going to have two. We're going to have flagella, and we're going to have cilia, right? So the flagella are also the structures of locomotion, but there's so many arrangements. I'm not going to talk about any of the arrangements. One of the more common arrangements are both of these cells, right? This is an algae, that's a protozoan, and this is a protozoan, right? But they have a biflagellated arrangement for their flagella. Here's the nucleus, right? And then they have two flagella. Okay? They are the structures of the locomotion, right? They are attached to a basal body inside the cell, not the plasma membrane, right? And ATP powers them. Okay? And as somebody remind me, the flagella of prokaryotes move how? How do they move? How do they actuate?
Like a propeller
Like a
on a
propeller?
plane.
Like a propeller. So they act like a propeller, right? With the eukaryotes, there's an extension and a contraction, right? And so they're much more elegant, right? And so this same theme is in a lot of eukaryotic cells. Think about jellyfish and the way they move, right? So there's a lot of different organisms that octopus move the same way, right? They extend and they contract, and they extend and they contract, and that moves them, right? So there's that same theme again. Like there's multiple themes going on, right? But these organisms all kind of have that same type of movement. Okay, good. So here you can see the flagella kind of move like an extension and a contraction, right? And then the cilia are kind of going to move similarly to the way that the prokaryotic flagella move. They undulate, right? And so what that means is that they move kind of like in a wave motion, right? And so there's kind of a wavy thing, right? The cilia are only found in eukaryotes. There will never be a cilia on a prokaryote. There will never be a fimbria in a eukaryote. Are you with me? So if you think about this image right here is showing a contrast microscope, and you can see the insides of a cell. But you can also see these structures that are found peritrichous to the cell. The cilia are always going to be found peritrichous to the cell. You can see them here, and you can see them there, right? And they move in a wave-like manner. So football's around. It's been here for a while now. It's the season. So put yourself at the 50-yard line, Cheska. And then it used to be a thing. I don't think it's anymore, but it used to be a thing where the audience would do the wave. Are you with me? So that someone would get up and they'd sit back down, and it had this undulating motion all over the audience, right? So that's the way these cilia move, and they move because of that. All these things moving all at once, these little organisms can boogie. They can move really fast, right? Much faster than those organisms that are flagellated. Okay? Any questions? All right. So
So just to reiterate, the cilia would be only like in animal cells opposed to the fimbriae that's going to be in bacterial cells, right?
correct. So also, Cindy, ciliated cells can be in protozoans too, right? As you remember from our lab. Okay. So Cindy, is it the left or the right? Which one is a bacterial cell?
The right,
Why is that, Cindy?
because it has the flagella and those little red things.
Those little red things are what?
I think fimbriae.
Starts Fimbri. That's correct. So you can see that they have these small projections here, right? Those are Fimbri, right? And then they have flagella. Eukaryotic cells will have cilia or flagella, but not both. Okay? Any questions? It's supposed to be an A right there. Okay? Any questions? Okay. All right, organelles. So there's a lot of them. I'm only going to hold you because we're looking at the structure and the functions of the cells, right? So I'm not going to expect you to know every single organelle. That's just mean. I'm gonna ask you to know these 6, right? So the nucleus, right? The endoplasmic reticulum, the smooth and the rough, the ribosome, the Golgi apparatus, and the mitochondria, right? Are you all comfortable with what these structures are? Does anybody need to go over these, right? Is everybody comfortable what they are?
Maybe
Can we go over with them?
the
Okay,
Or
let's go
Golgi
over them.
and
Okay, let's go over them. Nucleus, it's found in usually around the center of the cell, right? It depends. It can be moved, right? But it is the organelle that houses the DNA, and it also is the organelle that facilitates mitosis and meiosis. Okay. There's a lot of other things that happen in there too, right? But those are the ones I want you to know. Endoplasmic reticulum, the smooth endoplasmic reticulum, is associated around the endomembrane system. So the endomembrane system is the nucleus, it's the endoplasmic reticulum, both the smooth and the rough, and it's the Golgi apparatus, right? But the smooth endoplasmic reticulum is used to make certain types of lipids, okay? Rough endoplasmic reticulum is found right next to the nucleus because it's going to receive RNA, messenger RNA from it. And that, along with the ribosome, the rough endoplasmic reticulum and the ribosome, is used to make proteins. Okay. The Golgi apparatus is going to be found adjacent to the rough endoplasmic reticulum. It's right here, right? So here is the RER, the rough endoplasmic reticulum. It's studded with the ribosome, and then this is the Golgi right here. The Golgi receives the proteins and vesicles from the rough endoplasmic reticulum, and then it enzymatically modifies them, right? And then once it comes out of the very end of the Golgi apparatus, there's some vessels that will have the vesicles that have the protein in them, and those proteins are shuttled to other parts of the body, other parts of the cell that need them, or they're shuttled outside to be removed because they don't need them. Okay. And then the mitochondria are found at different parts of the cell. There are many of them in the cell, and this is where most of the ATP is formed, right? It's where the electron transport system is located. Okay, so all of this is described for you in the next few slides and a lot of detail, but I will ask you questions over what I just told you, and only on those six organelles and nothing else. Okay, so the last thing we'll talk about today before we take a break is endosymbiotic theory. How many people have ever heard of endosymbiotic theory? How many people have ever heard of it? Endosymbiotic theory. So theory in science means what? Miss Kayla. Anybody can answer.
inside.
It is. What's that? Oh, no, it's
Inside.
okay. Inside, endo means inside. Symbiotic means organisms in a relationship. Okay. So what we believe, theory, everyday theory is a guess, right? There's no data that backs up. Scientific theory is different. Scientific theory means that there's a lot of data that supports what we're saying, right? And there's a ton of data, molecular data, morphological data, all kinds of stuff that support this, right? And so endosymbiotic theory basically says that early on, we derive from archaea, right? We know that already, right? We talked about that phylogenetically when we talked about phylogeny about a week ago. But what we basically know is that within the symbiosis, there was two organisms that were living together, right? And so here's an example, right? So you had early eukaryotic cell, right? That was anaerobic, and it engulfed a bacterium. And then over a period of time, the bacterium was aerobic. It essentially started to only do things like cellular respiration, right? So on this top image right here, you can see that there is an aerobic bacterium. That means it uses oxygen to make energy, and an anaerobic eukaryote a long time ago only could live in where there was no oxygen. They came together, but over a period of time, this bacterium stopped functioning like a bacterium, and it became specialized. It became our mitochondria, right? And so the the current eukaryotic cells are really the result of this symbiotic relationship over millions of years, right? Where the bacterial cell became the mitochondria. And so how do we know that? Well, the structure of the mitochondria is just like the bacteria, right? Its own plasma membrane. It has Mitochondria have their own DNA. It's one chromosome, right? And if we take a look at that DNA and compare it to bacteria, it's very, very structurally similar in the way it's put together to an archaea, right? And so where we came from, where our cells came from, where eukaryotic cells came from, was a symbiotic relationship from an early eukaryotic cell and an aerobic bacterium. And then over a period of time, these aerobic bacteria became the mitochondria. The same kind of thing happened in algae and in plants, right? Where the cyanobacteria, right, became really the chloroplast in plant cells, right? And so it's an important thing to consider how these things work. There's a TED Talk you can watch if you want to learn more about it, but I'm only going to be testing you over what endosymbiotic theory states, and that's it. Okay? Is there any questions about what we talked about? So there's a couple of charts here for you to look at if you want, but I am done with prokaryotic and eukaryotic structure and function. The only thing we have left to do is to talk about cellular respiration, and we will do that on Thursday. And so our first exam is next Thursday. Our first lecture exam is next Thursday. This Thursday at 10:30 is your first practical. Cindy.
So, yeah, so you were saying, okay, to go back to the endosymbiotic.
Yep.
You said that we were going to be tested only on the fact that, I guess, the eukaryotic cell, the mitochondria came from the bacterium.
From bacteria. That's correct.
Okay. So basically, that's kind of what you just.
Here's in a couple of sentences. Are you with me?
Yeah.
Aerobic bacterial cell combines with an anaerobic eukaryotic cell. Million years later, you have very complex cells that are aerobic, and the aerobic bacterium became the mitochondria. That's it. Okay, Cindy?
Okay. And I saw you had where did you get that little slide from? I don't see it.
What little slide?
The slide that had that little anaerobic bacterium cell.
So I just added it this morning. You know, when I get up at
Okay.
I get up at 5:30 in the morning, and I think, well, you know, that's going to be hard to explain. Let me add some slides. And so what I'll do is I will put those on for you. Okay. I will put I'll put the new slides I put into on Blackboard for you. I'll do that right now while we're taking a break. Fair enough?
Yeah, thank you. Because I was like, where is that slide? I'm trying to follow.
Because you know, when I'm reflecting on what I do, I'm like, this might be easier if I do this. So
No, it's okay. I just want to make sure that I got it right.
Okay.
Thanks.
Anybody else? Anybody else have questions? Cheska.
Yes, no, I don't.
What's your question? You don't have a question?
No.
Okay. Anybody else have questions? Okay. So it is 12 o'clock, right? Why don't we take 10 minutes, take a little break, come back under lab and we'll review for the practical. Okay? I'll see you guys at 12:10.
And that YouTube link that you sent is going to be on that slide too, right? Sorry, just want to make sure.
Yes, it will be, I promise. I'm gonna do it right now, okay, Cindy, and you can check it in 2 minutes. Give me 2 minutes, okay? And I'll fix it. Okay. I'll see y'all in just a little bit. I better put that on right now. Cindy's gonna kill me.